Hi everyone, my name is Ümran Yaman and I am a PhD candidate at Hardy Lab at UK Dementia Research Institute at University College London. Today I will be presenting a summary of our work with Oxford Nanopore long-read RNA sequencing. In Alzheimer's disease, genome-wide association studies have identified key genetic risk variants, which are mainly microglial. However, the function of GWAS risk variants are still unclear in disease. In this study, we have performed long-read RNA sequencing in 9 months of age APP and LGF knock-in mouse model to identify changes in splicing and novel transcript isoforms in response to amyloid load. By 9 months, we do have a microgliosis, amyloid load, but no cognitive impairment in this model. For this experiment, we have used bulk tissue from cortex and used PCR cDNA sequencing kit, specifically SQK BCB109 barcoding kit, and we have used ProMFIN 48 flow cell with R941 chemistry. We have six replicates in each genotype, and we have controlled for sex and possible batch effects due to pooling or sequencing. For the data analysis, I have used Oxford Nanopore pipeline for pre-processing the data set from each mouse, and I have collapsed all the detected isoform with string type. Then I have used isoannote from functional isotranscriptomics framework to further annotate the isoforms, but also continue downstream analysis with isoform switch analyzer and DXig for differential isoform usage and isoform switch. To further annotate the dataset and isoforms, I have used external databases and I have used the inputs for the isoform switch analyzer. And these external databases including coding potential and protein predictions. Briefly, isoform usage and isoform switch events are measuring the proportion and the direction of expression changes on isoform level per gene, thus not biased through the cell proportion. And alternative splicing is a process of producing variably spliced mRNAs from a gene, and we know that it plays a critical role in the regulation of these transcript expressions, and it leads to protein diversity. We have profiled both isoform usage splicing and isoform switch events genome-wide in APP and LGF compared to control. Our initial results show that we have isoform alterations, such as isoform usage and isoform switch in some of the disease-associated microglial genes alongside other cell types, including neurons. This particular gene, CAPG or capin-actin protein, is previously found to be enriched in plaques in integrated human Alzheimer's disease omics datasets, and also is found to be enriched in amyloid response in mouse model. Here, CAPG specifically belongs to disease-associated microglial subset. What has been missed in the studies is that the specific isoform of CAPG, which is the CAPG202, and the novel isoform of this gene is increased in the same direction, and CAPG201 and 203, first and the third isoforms, are decreased in response to amyloid as opposed to this. Thus, a CAPG having an isoform switch event, which is a specific pattern of isoform expression. Other genes having a similar pattern includes lysosomal genes and other disease-associated microglial genes such as SYNGR, synaptic green one. But also interestingly, we have detected some pseudogenes. Also, a number of isoform usage and isoform switch events are occurring in novel isoform of both microglial and neuronal genes as well. Other important points are that the well-known GWAS risk variants such as APP and MAPT did not have isoform usage, isoform switch events, or if splicing. But we have captured this microglial enrichment and some of the neuronal genes. And there are other minor differences between two sexes. If of interest, please don't hesitate to contact about them. In addition, we have identified the genome-wide splicing profile using PSI Sigma, which is a splicing tool developed for both short and long-read RNA sequencing. PSI Sigma also detects the mutually exclusive exons, which is not possible to detect with the other splicing tools. Here we have identified around 100 genes displaying one or more of these classes of the alternative splicing, and these are occurring in APP and LGF compared to control. Here, the gene list includes clathrin light chain A and some of the disease-associated microglial genes again, and some AD risk genes such as clustrin. These genes collectively found to be enriched for functions primarily associated with endomembrane system organization, actin filament binding, and actin cytoskeleton reorganization, and they are related to pre- and postsynaptic neuronal functions, regulating the shape of neuron or neuronal projection. Transcript isoform modifications exhibit enrichment across various cell types in the brain. These include border-associated cells, oligos, microglial cells, and neurons. A particularly interesting aspect is the enrichment of the genes with isoform level changes associated with the diverse synaptic pathways, which is unseen on the gene level. Thus, we hypothesize that the synaptic changes are occurring in response to relatively early amyloid load, is protective response, and is a part of the amyloid cascade. Our results suggest that the concurrent alterations in the synaptic physiology alongside the upregulation of the AD risk genes in microglial proliferation and activation during the initial stages of the disease progression aligns with the earliest cognitive changes. With long read, we have now created a more comprehensive picture of isoform-specific indicators holding the potential to provide information about the disease stage. And this is independent of the gene level expression changes resulting from the cell proliferation. Lastly, I would like to thank all the collaborators and the audience for the listening. Thank you.